Thanks, Jeremy. Good morning, everybody. Um, Actually, yes, Jeremy is absolutely right. This is a different kind of presentation. It's a majority of these are just my own thoughts, some questions that I'm posing to us as a community to think, ponder, and hopefully together find some solutions. But I have to give credit to Nikolai actually for sowing the seeds of this presentation in my mind when we met at AAPS in May. I don't know if you remember, but we had a very nice dinner. Definitely wine was involved, and maybe that's what really got our thoughts flowing. Uh, but the next day, when I was really trying to recollect this conversation, Nikolai had provided lots of ideas, thoughts, and I don't know how many of you have talked to him when he really gets passionate about something. He just flows with ideas. But what I did remember is his style of asking, ask yourself this question, ask yourself this question. And from that time onwards till now, that's really what I did. I asked myself a lot of questions of what is really bothering us in this predictive immunogenicity realm. And what I'm presenting here are some of the issues and potentially solutions, but the solutions are not vetted, they are not validated, and they definitely don't represent any particular opinion or recommendations per se. So please take that with a pinch of salt. But the key is that if these elicit a discussion at this platform, and then together if we can build on it, improve that, that would be really fantastic, and then I would have kind of achieved my goal. So the title of my talk is Working Towards Building a Value-Added Proposition for Immunogenicity Prediction and Risk Management. As you all know, there are several causes of immunogenicity. Some of these are product-related factors, and some of these are host or patient-related factors. I don't really have to go through this list. It has been mentioned repeatedly in several presentations, has been covered extensively in several publications. But just to name a few, we are looking at tools today and tomorrow that would be looking at sequence differences, potentially aggregation, host cell protein. All these contribute to immunogenicity. The host and patient related factors are equally important, and it is important to know and understand what is the immune status of a patient, whether the patient is undergoing a autoimmune response, infection, inflammation, how do they contribute to the initiation of immunogenicity, and last but not the least, the particular haplotype of a given patient and its consequences on immunogenicity. But the key is that based on all this information, so we have really extensively worked on all aspects of the previous slide, and the work is still going on in better characterizing all those risk factors. But as this knowledge is coming in, certain paradigms are being constructed, and some of them are that there is a general impression out there that the in silico and in vitro methods, they are available, but the predictive power is limited. Now, true or false, this may be working in certain situations really nicely, but in other situations, we also know that it has failed miserably. And that doesn't lead to a confidence building phenomena for these tools. So then people have started looking at animal models. Now, animal models do provide an in vivo perspective, which is a little better than the in vitro methods, but they clearly do not reflect what is happening in humans. So even after the results that you get from in vivo models, you're left with the question that do we have the 100% certainty of going into the clinic with that information? So now the paradigm that is evolving is that there definitely are several factors that probably should not be looked at in isolation. They probably should be looked in terms of how they work together to cause immunogenicity. These multiple factors that are contributing to immunogenicity should probably be studied in concert. So experiments, clinical trials should be designed in such a way that we have a sensitivity analysis to look at the effect of each of these factors, not individually, but together. So current strategies and tools address one risk factor at a time, and that is probably one of the issues that we are encountering with, which may not be giving us the type of relevancy with the clinic, or which is probably not giving us the specificity that we are looking for. So we need to measure and assess the cumulative effect of these multiple factors, and that seems to be a paradigm that is evolving lately. Finally, can predictive immunogenicity be a population-based science? And which it is to a certain extent right now. But I would actually like to take this forward to see if the information we have right now can actually guide us to making predictive immunogenicity more similar to the personalized medicine-like approach that we have seen for biomarkers. And to this last point, I have some slides to the end of my talk which will elaborate on that issue. And I would actually like to open this concept to all of us, whether this is a paradigm shift worth considering. In this slide, what you can see is what we have been doing so far. We do the in silico predictions for the biotherapeutics. Then we have a bunch of in vitro confirmation tests. This is just symbolizing the MHC peptide binding. Please don't worry about the lack of focus on these images. It's very deliberate. I really don't want you guys to pay attention to the pictures per se, but it's just a flow of concept. And then we do the ex vivo confirmations of T-cell assays, APC assays, and all sorts of things. And this is what I'm calling as a loop. Now, after the set of these experiments are completed, some of us actually do take efforts to de-immunize those peptides that have been identified and, so to say, validated by these in vitro methods. And after that de-immunization, there is a question mark in everybody's mind. Do we then go directly from here to the clinic, or do we bring it back into this loop to then reconfirm the observations that we have made in vitro? And this is where there seems to be a confusion and a lack of clarity in a majority of people's mind, is that how many times do we follow this loop to get that 100% specificity? Now, I have been talking to not only my network in biopharmaceutical company in building all these concepts through the talk, but I have also been talking to the CROs. So I have figured out that the average tag price of going through this loop is at or around 500K. And Jeremy and Nicola, you guys can correct that figure if I'm off. But that is generally the estimate. Now, if you look at this in terms of the value that we are trying to look for this effort, the questions that really need clarity is how many of these loops are necessary, what is the time and cost that are required for each of these loops, and how can this be effectively introduced to into that very tight development timelines that we all face. Honestly, when I took a look at this timeline and these efforts in the extremely fast-paced timelines we have at Regeneron, for some programs it was absolutely next to impossible to even consider doing anything like this. We are a company that develops completely human antibodies, and in which case it's a fight, completely an uphill task to convince the senior management to even get into an effort like this. It may be a little better of a sell when we have compounds that are high risk and probably for life-threatening disorders. But just to give you a perspective, these are the questions and issues that we are dealing with right now. The key here is the validation of the clinical results and its utility. So after doing all this, if we are not certain that this effort really translated into clinical results that were safe and efficacious for the patient, then this value is lost. And that's what I have tried my level best to concentrate on for this talk. But are we just sitting twiddling our thumbs? No, there are lots of efforts going around in the international community, and a major effort based on Bonnie's leadership has been started with the Therapeutic Protein Immunogenicity Focus Group. And we had a nice survey that was actually conducted last year. And this slide is actually just one summary slide of the key observations that we made. The observations were that people felt there are several gaps in our understanding. Some of them are that the proof of concept or the validation of these tools is missing, and that does not allow them the required confidence to invest in these technologies. Now, because there is no investment, there is no data created, no data in the public domain, there definitely is a clear lack of benchmarking of these tools. So if there was a small to mid-sized company, they really don't have resources to go around on a fishing expedition, they would really like to just go to a paper that says, this is good. Follow this and you will get so much sensitivity and so much specificity. That is what a majority of people are looking forward to, and I think there are conversations happening where something will translate hopefully soon in our area. Then there clearly is no database for making prediction correlations. There are databases, however, those databases for some reason that I think are obvious in some terms are really not taken with that level of confidence that they should, because the databases themselves are evolving. Now we really don't know how much those databases should really evolve in order to reach that specificity level that we want or we feel comfortable with. And then there is a huge impression about the cost. Now here I have starred, I think it is this column, that doesn't appear that cost is a major concern, but when I actually extended that question through my own personal communications, in most people's mind, the cost is definitely a concern, which without proper understanding at the very senior management level, this effort is completely discarded. Oh, this is too much, it will cost too much, and I don't think it will lead to any value. So that's the impression that is out there. Now, okay, we found out that through this survey effort, the gaps are there, but then I really started digging deep into why these gaps are the way they are. What were the reasons this gap developed in the first place? And I was saying the key is lack of confidence in the value proposition. People don't seem to have realized what is the value of doing that. So if we just concentrate on that for a minute, the question is, even if T-cell epitopes are found and muted, then does that translate to no immunogenicity product? That is a very typical senior VP and above question. If they want to invest, they want some sort of 100% specificity. I don't know what really is the premise for an unrealistic expectation like that, but this is something we are fighting with on a daily basis. And we just had some conversations, and you guys also seem to be experiencing the same thing at Pfizer. At present, lack of predictive immunogenicity information or even unfavorable results, and that is key, is not a blocking for licensing. So you can easily license a product, especially a monoclonal antibody product, without any predictive data. And even if you have done predictive data which has failed to give any correlation, that doesn't increase your risk for getting licensing. So then, for the people who are managing the risk and cost estimates in your companies, this is a no-brainer. Why should we invest in this? Finally, familiarity of tools is there, which is what we gathered from survey and all sorts of personal communications. The familiarity is there, but people are not using them. Now, there are several aspects to that. Sometimes it is mere accessibility, and sometimes it's these impressions that I'm talking about. Now, accessibility is a relatively easy problem to resolve. I think ProImmune has done a fantastic job in communicating through their webinars, their emails and presentations. But are there other vendors out there? Are they reaching to everyone, what I would call as a key stakeholder in the entire biotech pharma industry, so that at least the accessibility issue can be dealt with? And what are sponsors getting for results in terms of success and failure? We really are in a completely gray area. I mean, we each in our own company have our own observations and things like that, but we are not sharing them. There's not much information about the failures especially are coming in the public domain, as a result of which the science has stopped to evolve. As you see, for all disciplines, scientific disciplines, positive and negative results are reported, and it is based on those negative results that we really quickly find out what doesn't work, and then efforts are focused and concentrated, and they evolve in what works, and that is what is missing here. So one of the major reasons we are in this state is because the legal component is a big impediment. When I was at Pfizer, we had the same conversations, the exact same thing I'm experiencing at Regeneron. I've heard the same thing from J&J and so many other companies, that there is a big issue about releasing especially the failure data in the public domain from the legal departments. And we have been seem to be stuck at this point, and really I don't have a clear solution right now unless we dig deep into what those issues are from the legal standpoint. But the time and cost is something that we can talk about, and something that I've thought a little deeper, which I'll present to you in my subsequent slides. So the cost, if I look at it, is about 500K for the entire loop, as I'm calling it, compared to a cost that I took just for our own clinical trials, about 55 million for a open label extended study of a reasonably sizable number of patients that is done on multiple components. Now, if you look at it, this particular effort is literally a drop in the bucket for the budget that goes in just a single trial. And how many trials do we do for a product? If you look for the entire, if you look for an entire cost of an entire program from preclinical to phase 3 clinical and beyond, this is literally nothing. They should not be even on the boards of consideration. So cost doesn't seem to be a issue. It is more an impression, and impressions can be dealt with by giving the right information to the right people. So the time is also not a big deal. If you look at it, if what I've heard from some of the CROs is that that loop can be completed in about 4 months. Now, if planned properly at the right juncture, whether it is at the preclinical stage or even as the clinical program is evolving at each and every phase, 4 months is definitely something that can be adjusted. But when I talk to several people from across coast to coast in the US, as well as 2 companies in the Europe, this is what I really got, which was very fascinating. The time it actually takes to get the job done in an organization in a product timeline, product development timeline, is actually 1.5 years. And the biggest issue is there are delays in the decision making in terms from the side of the sponsor. Months go to just figure out what to order and when to order. And even if it is ordered, for some reason there is probably fighting in the budgets and the dollars that are allocated, there is always a minimalistic approach that is taken. And that minimalistic approach doesn't end up giving a comprehensive data with which you can do anything meaningful. So at the end of it, it's a completely wasted effort and wasted time and money. The interesting thing is that people don't feel comfortable unless they have dabbled into it themselves. So everywhere there was a parallel internal effort of predictive immunogenicity and a CRO effort for the same program, with different questions being asked on different platforms without the right statistical controls. And then when both data come in, there is a tendency, and I'm not saying everybody does that, there is a tendency of putting these data sets together to figure out if they make sense. And that seems to be confounding for many people out there. And I think this is something that can be quite easily rectified if we look at it. So you may want to look into your organizations and see if something like this is happening. But this is clearly happening. So the timelines then progress, and it leads to eventually a total frustration on the minds of the stakeholders, who continue to lose that kind of confidence in these technologies. And make a note right here, there is absolutely no science playing a part. It's all impressions, it's all observations, and it's things that are not done the so-called right way. In summary, a lack of clear understanding of the risk-benefit ratio to the value-added proposition that I'm talking about. This value-added proposition tool actually, this term actually came in one of our TPIFG telecons, where we were discussing a whole bunch of things, all these gaps, and it was making me uncomfortable that the conversation isn't going anywhere, and it dawned on me eventually that there is no value. We haven't really figured out what is the value. I think if we align with a clear value set, everything else will get aligned relatively easily. Measures taken by the international community to address these gaps. As I was saying, there are different people who are working on different various angles, and clearly these are the 3 groups that you are all familiar with. They have done a lot of work in this area, and hopefully these results will start trickling in. The key objectives that they are trying to address is there is a need that they have identified to select a few methods or few tools that would be validated and hopefully reproducible that people can use consistently. And they're trying to demonstrate the human or clinical relevance of some of these methods. It's not a comprehensive proposal, but it is definitely something that certain tools will definitely get addressed the right way. The gaps that I feel still need to be addressed is that we need to develop industry standards, and that is where I think the community can come together, the experts from various organizations can come together to create recommendations for those who may not be that advanced in their organizations in predictive fields of immunogenicity. So to those, if we can provide some recommendations based on the knowledge we have right now, that could be done through sharing of experiences or even using some of the internal data, but to show them some sort of a clinical relevancy of these tools. Then more data should come in the public domain. I've touched on this before. The risk factors have been identified, but the understanding to the extent of the influence of each of these factors in concert is still not well developed. And whenever we start addressing these gaps, if we miss on this point, we may be back to square one, because I think efforts moving forward should be looking at risk factors in concert, or if not all the factors together, at least those factors that are most clinically relevant to a given product and a given indication. The key is then to increase the confidence in the decision making and predicting relevance in the context of the biotherapeutic and its specific indication. And it is this point that I'm trying to build on in my next few slides. Let's see what sort of information we can provide to the senior management by which we can possibly get a buy-in for their investments. So I do all sorts of ad hoc reading, and I was looking. So I knew there was a risk. So prior to this slide, before this slide even came into my thoughts, I knew we were dealing with a certain amount of risk. We were dealing with a certain amount of investment, and clearly there was going to be some outcome. Now these three terms are very popular in a couple of industries. One is insurance industry and one is the stock market. So I did a whole bunch of non-specific reading in both of these industries to see if there are any papers that would give me some thoughts. But most of it was so much of a technical jargon that I really couldn't even digest. But then I bumped into this article which was on oil drilling, and it suddenly occurred to me that this actually has a very nice, better analogy. So let's take a little diversion, but let me come to this and tie it up with our immunogenicity problem. So in the oil drilling industry, they have predictive tools, and the analogy is quite parallel and similar. So this is a ship that is trying to locate where the oil depots are in the underground basins. And what they do is, besides using some gravitometers, magnetometers and all that, within the gravitational field, they see fluctuations, which is how they can figure out where the oil is. But they have then evolved a technology which uses a sensor which creates a shock wave in the water, and those shock waves travel and they reflect and they are picked up by these sensors. That gives them an accurate location of where the oil wells and the oil fields are. However, apparently that's not enough. That cuts down the risk of the costs it would incur to go and drill a well. But there are other risks that are involved, and those risks are, this is an oil field or an oil bed as they call it. Along this oil bed, there are several pressure points. And if you don't know where you are puncturing that particular well, you have a huge environmental risk at your hand, the costs of which are enormous, and I think we dealt with a couple of examples in the last 5 years. So the risks are environmental, as opposed to what we are dealing with is the patient health and the safety, or even the efficacy, I would say. And the costs are huge in penalties for this industry, and the costs could be equally huge for us also. We haven't really done a measure to figure out how much those costs are. So the challenges are very similar. And it is with this that I thought, from these experiences, the oil well drilling industry actually learned their lessons through these accidents to learn to invest into these new technologies, wherein using these kind of scanners, they figure out exactly where these pressure points are. The blue well is something that has used these pressure points, and the red well is a well that probably didn't use it, and then definitely they had an accident at this point. The incurred costs they calculated for this industry were 50 to 100-fold more than what it would have been if they would have put those costs in the prediction. Now, if you look at this analogy in our business, are we waiting for a catastrophe to happen to really start learning the value of prediction and predictability of immunogenicity and be aligned to have those investments in place? So the learnings for me at least was that we need to build a case study of systematically calculated cost basis of immunogenicity prediction versus not doing it. And that can be done not through just mere investments, but there are some proposals I'm putting forth, right or wrong, we can all discuss. But I thought it was an interesting approach that we can pursue. So see what happens in the next couple of slides. Building a value-added proposition. In this slide, I'm looking at the impact of immunogenicity prediction that goes really well beyond the realm of science and starts delving into economics of immunogenicity. At this moment, what's happening? Risk management is being talked about, and risk management, in my mind, should really be a good balance between prediction and mitigation. But just as we have evolved as an industry, our emphasis is overtly on mitigation as compared to prediction. There is very little prediction in most programs that I see as compared to mitigation. And somehow this business model has survived. Now that is the key. If this business model has survived, it will take a lot of effort to change the direction of this dogma in the other direction, or even to get it all properly balanced. But there are some ways, some questions that can be asked to highlight these points. Let's forget the pharmaceutical industry for a minute, but we are all part of a healthcare system, irrespective of what country you're looking at. So if I just look at this one publication, which is the only publication I could find, which was talking about the healthcare burden that is related to immunogenicity. Now, in the Factor 8 arena, the UK Haemophilia Centre Director Organization has made some recommendations in this paper, and they were very interesting. They have recommended that in order to manage the bleeding episodes of some of these people who develop the inhibitors, you have to give these agents that are called bypassing agents a certain number of times. When they did a meta-analysis of the costs that are incurred for that healthcare system, it was quite eye-opening. On an average, there are 2 to 3 doses needed of these bypassing agents for a complete resolution in a single patient. And these average costs are about $161,000 per year per patient. Now, these are just average costs, but when they actually took a cohort of 14 patients who may have varying episodes in their 1-year time frame, then they realized those costs could actually escalate quickly up to 10 million per year for that simple cohort of 14 patients. Now translate that cost on a worldwide basis. That's a huge number, all arising out of immunogenicity, all irrespective of the fact of a very sophisticated mitigation strategy that we think is working, but at the back end is creating this huge burden that we have to be responsible for sooner or later. So park that information on your mind, and let's turn our attention to what's happening in each and every pharmaceutical company. Now, we have immunogenicity. It leads to either safety or lack of efficacy. So what are the drug costs that are being incurred by the company in clinical trials? What are the hospitalization costs? What are the reimbursements costs, if there are any? And what is all this translating to in terms of the wasted resources of the organization in light of an underlying immunogenicity problem? And is this something that can be resolved by putting a little bit of time and little bit of efforts in investing in predictive immunogenicity? That is where I'm trying to drive this conversation. So the costs of mitigation strategies that have been employed to manage these adverse events are what is really at the crux of this whole equation. There is an impression that the mitigation strategies are working, but what we need to do is put that in perspective with the costs that are being incurred. So advanced products, the other problem we have is everybody is looking right now at the regulatory bodies to give us some sort of a recommendation or guidance as to should we use it or not. And even in the senior management circuits, you will often see that if the FDA is not asking, the EMEA is not asking, why should we bother doing it? It's almost like people are waiting for the regulatory agencies to come up with a docket that here, we want this, otherwise you will not be licensed, and then everybody is willing to jump in. So the problem we have right now is the advanced products that are already out there in the market, they have evolved completely without any predictive analysis, and they are reasonably successful in the market. My next slide will actually be highlighting that issue. They are so successful that there is really no initiative or no drive to put any additional investment of time or money. The current risk management strategies are reasonably successful and they provide a desirable outcome. So even in the face of immunogenicity for products like Remicade or Humira, then it's all a game of numbers. It's like how many people are not getting efficacy, and that's okay because there are so many others who are getting efficacy. We are getting the billions of dollars that we want. The bottom line of the pharmaceutical industry is looking good. So why change course? Why change direction? And for some reason, there is an impression that there is a lot we have to do to change this course, which really is not. So if value-added proposition is what we are looking at, and all that information that I gave you are the type of questions we can ask, then how do we project this information? Here is a simple pie chart of what I got from, I think, just through Google. It's a 2008 arthritis drug market portfolio, in which, as you can see here, Abbott's Humira is at 4.5 billion. At that time, it was around 28 to 30% of the market share already. The only difference, or one of the major differences, it was a human antibody with a perceived low rate of immunogenicity at that time. Now, the key is, I checked through some of our marketing folks, Abbott Humira has actually touched 9 billion or will touch 9 billion by the end of 2012. That's a twofold growth, significant growth in a product that is ever seen in the biopharmaceutical industry. And if this is what we are dealing with, there is a lot at stakes for even the senior management to consider some of these questions that I have posed. Humira is 50% of all of Abbott's drug sale. That was a big, big number. Now look at the risk that is being assumed by the senior management for these kind of revenues that are already under a huge pressure. The pressures on Humira sales are coming from a patent expiry that is at or around 2 years. Now, I couldn't get an exact date, but I know it's in the neighborhood of 2 years. Then there is a very, very strong competition coming from small molecules, which are providing the oral alternatives, and that is creating a market erosion, or there is projecting to be a market erosion. And then there are definitely some biobetters in the competition that are really on the heels, and some of them have just entered phase 3 as well. So with that kind of revenue, that kind of dependency, is this company really, is it even to take the risk of not improving on the immunogenicity factor, which seems to be the single most differentiating factor that is coming between them and the biobetters? How can we ignore the issue of predictive immunogenicity? And these biobetters that I was talking about, here is just an example, and it is all in the press. It literally came about 2.5 months ago. The role of immunogenicity as a powerful differentiator in this equation is what I want to emphasize. Humira label was saying that it is 1 to 12% NAP positive in a 1-year study, and right now the literature is actually projecting at around 35%. Of these, only 4% of these NAP positives are ever reaching DAS28 remission, so they have to forcibly take a switch on the biotherapeutic that they are being treated with. So that's a loss. That's a loss to the clinical trials as well as a loss to the company. Now, Ablinks, on the other hand, I've been following their ATN-103, which is ozorelumab compound. It's basically a small nanobody, about 15 to 20 kD. And they have done something to even improve the PK, so it has a reasonable PK profile. But they just came up with their total composite result of the open label extended study, and they only, one of the key figures they really highlighted was a 0.75% NAB at the end of study of 1 year. So if you compare that number with this number, which potentially can be even higher if the numbers are really in this category, then this is a significantly better product. And the differentiation is happening with nothing else, absolutely nothing else, but only immunogenicity, because the PK is comparable and immaterial. Safety and efficacy, the efficacy is quite equivalent, and in fact at some point I saw that it was 70% reduction was happening in 3 months. Now is that a contribution of immunogenicity or lack thereof? Is something that should be considered. So this is how I feel we can slowly and steadily build a case for predictive immunogenicity as a value-added proposition. This is not something that can be ignored or kept on the wayside anymore. The business concept that I see emerging is that there are several companies out there, and I actually am collaborating with some of them. They don't have the resources like the J&Js and the Pfizers. They don't have anything to do de novo, but they have access to technologies by which they can quickly jump into the product pipeline. And one of the things that is emerging is, if you have a drug like Humira, which has already established a huge market space of about 9 billion, I mean, that's a huge number. We can take a significant pie out of this by just coming up with a quick compound that is just better in immunogenicity. A very easy, addressable alternative for them without having to do the research of a target, without having for them to do any research of a clinical comparability, because they don't have to be biosimilars, they can just be biobetters and quickly come in here in about 3 to 5 years to take a big chunk of this pie. That is something the big pharma is soon going to pay attention to. This is where I'm seeing the value proposition evolving, not just from a scientific point of view, but also from an investment point of view. So these are definitely, so as we are talking about all this, efforts have to go into this. And these predictive immunogenicity efforts is what I'm now giving a tag price of the value-added proposition. So some of you may have known that in 2004, FDA had a critical path initiative, and they wanted the agency and the industry to work together to actually figure out exactly how much does it cost to develop drugs, both small molecules as well as biotherapeutics. So I came across a very interesting study that's in this paper, and I figured out that there were 17 biotherapeutics on which they had done a systematic cost analysis of these biotherapeutics. Now the database actually consists of 522 biotherapeutics that were combined to have a total between recombinant proteins as well as monoclonal antibodies, and these were products that had reached an FIH stage by around the span of 1990 to 2003. The interesting thing I thought really in this paper was they took into account not just the approved products, but they also took those that were terminated. And this is what is very interesting to me. The terminated products is what they calculated as a cost basis. So how much did you lose because you lost a product to immunogenicity? Now, parameters that they looked at was the development time, the success rate, the phase transition going from one clinical phase to the next, and the out-of-pocket costs as well as the cost of the development failures. And this database, I don't know, I really didn't access this, but I think it should be publicly available. It is available at Tufts. And there are some key highlights to this. They calculated the costs that are taken for all these clinical phases, and really if this is not clearly visible to you, that's okay. The key number is that your total out-of-pocket cost for a particular biotherapeutic is about 169 million at $2,005. So now this number I'm projecting easily about about 500 million at least at today's dollars. Now with that number, when you take at the delays that occur during development, there is a factor of time that gets multiplied to these out-of-pocket costs. So out-of-pocket cost multiplied by time is what gives you the capitalized cost for the development of a product. And that was a staggering 1.2 billion in 2005 dollars, which is definitely much, much more. So look at the risks that we are sitting on and look at what is at stake. If this is the number that you look at, that 500K of just that single loop, is infinitesimally small, and I then really don't see why there is so much issue, why there is so much uphill task of fighting back and forth in budgets and investments. Now, in terms of the the key factor that I learned here is calculating this cost. They are what are called as phase transition probabilities, and they are done by a very simple formula. How many number of compounds actually progressed from one phase to the next upon the total number of programs that actually were considered for that analysis that progressed or failed. And that gives you the phase transition probability. If you take that phase transition probability, it actually goes into constructing what is called as the clinical approval success rate. Now, we may not be aware, and I was not aware, but every finance department of small and big pharma has to track that figure because, and I'll show you why. This clinical approval success rate in this publication and this study for all those compounds taken together came to a mere 30%. So look at the numbers and look at the downfall. $1.2 billion in 2005 for a single program. And out of doing all these programs, you're only getting a 30% success rate. So a lot of water that is being put in is actually already leaking out in terms of the funds that are being put in and the resources that are being put in. Now, why is this number so important? It's important because this ASR is directly taken into consideration for calculating a very key figure called beta for any given company. The beta is the variance or the risk or the variability that one company has in its product pipeline. It is that risk that determines how much premium the investors, the mutual funds, are actually willing to pay as a result of this beta for a given company. So the company's job is to minimize the beta, maximize the probability of success, and thereby have a better risk premium so that people are willing to pay more for that stock. And all this then reflects into the stock price. But the key in this equation is I felt that immunogenicity can be a stepping stone in this sequence of stairs that lead up to the stock price. And immunogenicity actually contributes significantly in these equations that I just showed you about Humira and Ablinks to this beta factor. If immunogenicity can be controlled, it has a direct reflection eventually on the stock price and the way you can manage the risk profile in your product pipeline for biotherapeutic compounds. Now, all said and done, but again the question comes that I can say all this to our senior management and they would still probably not budge in their chairs because of all the perceptions that they have had in their mind for several years together. And in fact, I can even share this with you. Legal won't allow me to put it on the slides. But at Regeneron, I dug out a history that there was a compound called Axokyne. I don't know if any of you even heard about it. But there was a sophisticated, absolutely sophisticated, state-of-the-art predictive immunogenicity effort that was done for that compound. And they were so sure that there would be no immunogenicity whatsoever. And believe me, that compound got killed only for immunogenicity. Only for immunogenicity. And that organization has zero confidence in these tools. So it is very, very difficult for someone like me to bring in that. So what can I do? So this is the only thing that is probably completely construed and constructed in my own head. And I'm laying it out to you guys. This has not been published yet, and neither has it been presented anywhere. But it's a very nice concept. See what you make of it, and if there are any improvements to this, I would love to hear your thoughts and suggestions. How can we derive modeling to build this value proposition, right? So I will give you the concept in this small box over here by taking one risk factor, pre-existing ADAs, something that is more simpler to understand, has very limited variables. So I have taken that as an example. Now, if you take pre-existing ADA as a risk factor, in fact, let me change the term with all these economics reading. I don't look upon these as risk factors anymore. I look upon them as probability factors because probability factors are influencing the eventual outcome. And it is the outcome that I think we, as well as the senior management, will be interested. So if you take a particular probability factor, you have a choice. You either invest the time and money that is required to predict what is the value of that factor to influence your outcome, or you have a choice not to do anything about it and just run with the trial. Now, those two choices lead to what I call as the value proposition index. That's my own fancy term. But what it basically means is it's a fraction that is the cost incurred at that decision-making point upon the outcome probability. And this is simple. Now, this formula I know is, I have confirmed through our modelers that it is correct. And this approach also is not all that faulty as yet. So, using these simple cost parameters and the outcome probability at each of these steps, we can easily figure out what the outcome could be either prospectively or even retrospectively after you have got the clinical data, to really see how this model can be built. In simple terms, the costs associated with the decision of recruiting pre-existing ADAs positives without the predictive efforts would lead to this negative outcome, and you can figure out how much you have spent to get this outcome. As opposed to if you were to use the pre-existing ADA factor and eliminate this arm and only improve your probability of going in this direction, the chances of your positive outcome are increased and your costs are thereby decreased and you save a lot of money. That same simple formula or logic can be used to determine the incremental cost of predictive immunogenicity for a given drug indication treatment prospectively or retrospectively through a model that can be significantly of a higher dimension than this little box that I have presented here. And apparently, I later on learned from some of my modeling friends that it's not that big of a novel concept. There are already softwares that are existing that are called the decision analysis softwares. These softwares are already in play from airline industry to the stock market and God only knows where. So I think someone getting their hand on one of these should not be that complicated. But a model that is inclusive of all the risk factors and the probability factors that we have talked about would look something like this, wherein at each node you have the ability to take a decision, and at each node you have a full knowledge of how much you're going to invest and what your outcome could be. And then if you do a clinical trial and put that clinical trial data on a model like this, you can do sensitivity analysis to keep on improving this model. So these are some of the product related factors and patient related factors that can all go into this, and at a very, very high level, the output that I felt could be useful is to estimate the costs associated with the immunogenicity and treatment outcome. So how much are we spending with or without the predictive immunogenicity, and estimate the impact of the success or the failure influenced by the predictive efforts. In this case, what is really important to me is I would love to see that if someone does take the time of doing that loop and even doing the deimmunization and so-called improving on the biotherapeutic, does this value improve? Does this model predict a better outcome at a lower cost? And if we can provide a validated model to our senior management and the community at large that is reasonably reliable, I think that could be the beginning of a turning of a tide wherein these tools can probably be looked at with a little more confidence. So if you do have a model like this, now one question you may have is that, am I working on a model like this? Yes and no. We have the drive to do something like this, but right now we are looking for recruiting modelers. Work has not started, and I'm not sure if this would be even proprietary. Actually, one of the reasons I negotiated with our legal and management is that I would like to lay this down into the community so that we can work collaboratively on an effort like this. Because a model constructed in one company that's only working like ours on human monoclonal antibodies may absolutely not translate into another company that's only looking for recombinant proteins. So I think this effort needs to be beyond the borders of a given organization. But if a model like this was constructed, then there are several possibilities. We can find out, is there a correlation between a given factor with the outcome to the treatment and success and failure of this effort? How can we assess the impact of the net present value of the investment that goes in re-engineering the biotherapeutic? That is something I'm particularly interested in. And would an optimized biotherapeutic significantly lower the cost of immunogenicity management? Now, I'm not talking about risk here. I'm talking about the medications, the hospitalizations, and all those things that fall under the immunogenicity management issue. What are those costs, and can we minimize those? The other important thing that I came across, or we came across, was that you have so many factors that we are contending with right now, making the entire system very complicated and actually beyond the capabilities of the human brain. So for a given indication and a given biotherapeutic, do we really need to look at all of those? Will the model help us stratify those factors of relevance so that in a given indication and biotherapeutic, you only look at factors X to Z, and maybe they are the most relevant. So the model may actually help you eliminate those that are not relevant for the equation. Now, this is something we can definitely do at a cross-company level collaboration. Because it's a model, I'm sure it will continue to have uncertainties, but just as we have developed very sophisticated models in the PK analysis, and they have evolved over the years, something like this can evolve as well. But look, an effort like this will have minimum cost impact and even minimum resource impact. The threshold analysis is something that we can validate with our own clinical results that can be loaded into the system. But the bottom line is, I think a tool like this would allow us to make informed decisions of what makes sense in terms of the predictive immunogenicity arena. Now, this was a little effort. Up to this slide was an effort to communicate how we can build the value-added proposition and what are some of the ways or questions that we should ask to drive the community in that direction. But in spite of all that, deep down, I still don't feel totally comfortable, and I'll tell you why in my next few slides. The reason is this, that when we look at the product-related factors, they all seem to be manageable or predictable. But when it comes to the host-related factors, I feel there is still a whole bunch of stuff we really don't know, that even the most sophisticated models may not be able to capture everything. And what is bothering me is out here. The medical practice, as we know, is definitely has always been practiced as a population response driven medicine analysis. But if you look at a population that has been identified, has the same diagnosis and the same prescription is administered to them, you still end up having some of these 4 different outcomes. In some cases where it is safe and effective, in some whether it is neither, and in some it is not safe, but in some it is not effective. So can predictive immunogenicity tools and models, they can possibly drive a desirable outcome for safe and effective medicines when we look at this. Now, if we just look at this not in terms of the treatment outcome, but in terms of immunogenicity outcome, we can probably use all the tools at our disposal, all the models at our disposal to drive everything in this direction, or at least in this direction. However, what is the specificity that we can ever hope to achieve? That is what is bothering me. And this is the reason. If you look at the polymorphism that we all have, as you know, polymorphism at its base is defined as a DNA variation of just 1%. And SNPs that you all know is difference of a single nucleotide base. But the key is the SNPs can be either a synonymous or a non-synonymous. I'm sorry, synonymous or non-synonymous. And in synonymous changes, what happens is the nucleotide base changes, but the protein, the amino acid doesn't really change. So there is no change in the primary sequence. In a non-synonymous SNP, there is actually a change that happens in the primary sequence. Now, there is a lot of literature I found, and I'm sure you are all aware of, that the synonymous changes in the genetic code can actually affect protein expression levels as well as conformation in certain cases. Now, if we were to use our existing tools, they will actually tell you that there is no change in the sequence, which means the wild type and the drug is identical, which would allow us to conclude that there is no risk of immunogenicity. But if this statement is true, and which is what is evolving to be true, then we really don't have a handle on what can happen in vivo. And this is what makes me nervous. If you look at factor 8 as an example, which is probably so far by far the most well-characterized system for this particular phenomena, there are different haplotypes. It's really not important to have the numbers of the haplotype, but there are certain haplotypes that play a key role in the F8 immunogenicity phenomena, and they each are responding to, or those MHC molecules bind to certain changes in the factor 8 molecule that are represented over here. These are the two drugs that are actually given, and the problem that is encountered is as follows. The wild type drug that is available out there has a position which actually changes, which is part of a polymorphism in the entire human race. So different communities actually have this different representation at this allele, and now look what is happening. What we thought was a self-protein being administered to a human being, actually that single human being alone is recognizing that wild type drug as a foreign protein, which would lead to immunogenicity with absolutely no fault of the sponsor at all. And there are a whole bunch of cases that are diagnosed in this particular scenario of the F8 arena, and that is what is leading to lots of new questions, at least in my mind, in terms of how can we learn from these experiences to drive predictive immunogenicity to push it towards the 100% specificity that we are all looking at. And from this, what I believe, there are efforts that are already underway in which that people are trying to identify these epitopes that are likely going to be immunogenic in the entire population for F8. They're also trying to identify risk ethnicities and populations or even individuals that can be specially at risk before they're administered the drug. And this is what is evolving in the F8 arena, that there probably lies some potential in developing personalized therapeutics for counteracting immunogenicity. Now, these mismatches between the infused drug and the endogenous drug, if you look, they are elusive, but they may well be predictable risk factor of immunogenicity. So is this a risk factor that we can actually capture as a part of those other risk factors that we talked about to improve on the specificity further? So I don't know if at the horizon, if all goes well for us and the predictive immunogenicity community, is predictive immunogenicity moving towards a personalized medicine approach? And if that was the case, then all these tools that we have right now, and maybe even some new tools that would evolve, would all come into picture and would all become relevant. Because at this case, then you are having a panel that you are characterizing each and every patient before treating. I don't see something like this happening for monoclonal antibody products that are for very, very huge markets like arthritis or Crohn's disease, but it probably becomes very relevant if you're looking at enzyme replacement therapies or any substitution products where it is life-saving drug and it is also probably leads to a very catastrophic event. So I won't go over this, but the analysis can be done in a following manner: that what are the MHC class 2 alleles that the patient has, what foreign epitopes in the drug can be identified that would not sit well with this molecule. Are these molecules, MHC class 2 molecules, common? How common are they in the population? Now remember, MHC molecules themselves are very polymorphic. So it really has to be a one-on-one combination of the drug and the MHC haplotype to fine-tune this process. So it really becomes complicated as we go down. Are the foreign sequences generated by processing in the first place? So in silico, in vitro, we can come up with a bunch of peptides and make some conclusions, but when you change those mutations back into the wild type protein, the peptide may not be generated at all. So the MHC molecule is not even recognizing them. So these are all things that need to be worked out before we start developing a comprehensive model that takes into account all these issues. So to me, I'm leaving you with this question: is personalized predictive immunogenicity an overkill, or will it truly benefit the patient in a cost-effective manner? And that doesn't just hold true for a personalized approach. But even where we stand right now for the overall predictive immunogenicity approach, I personally see a lot of value. As much as there is a audience that is willing to listen, I would like to propose a very systematic approach of evolving these sciences, the data, as well as the communications. But here are some thoughts.
Great, thank you very much, Manoj.
Thank you.